Good afternoon. Exam 1 in the rear view mirror. A couple updates. We might have them done and posted tonight. Okay? There's just a lot of scores to enter. And then we got to make sure we don't misenter them because you'll freak out. And then we have a lot of extra work to do. So basically we have 350 times 2 because we have to kind of do it twice. So it just takes a little time. All right? We'll talk a little bit more Monday because you'll probably have the scores tonight or tomorrow. I'm guessing at the latest. And I've looked at the exam and I've already started kind of analyzing the data. So we'll talk more on Monday. I've talked with a couple of you individually, and I want you all to kind of be honest with yourselves when you get the grades back. If you felt like you put in the amount of time you felt was necessary to be comfortable taking the exam. Now's the time to do that. Okay, there's plenty of time for adjustments. What I mean by that is if you really kind of blew off exam 1, there's still 4 more exams. Some of you are like, oh! Yeah, we actually went over that in the syllabus, so let me repeat that. There are 4 more exams that are all worth the same amount of points as this first exam. And we've got quizzes, and we dropped the lowest too. So you're just trying to start to hit your cadence now. And I know the first exam is a little bit of, who is this guy? Never had him before. Is he a pushover? Are his exams difficult? How do I study? I know those are a lot of the questions, and now you got a sampling of what the exam looks like. But I'm going to tell you, there is still plenty of time to make adjustments. And I hope that you agree with me that my guidance of coming to lecture and watching the lectures again, that's why I take the time to post them, and looking at your lecture notes and asking questions about lecture material will prepare you for the exam. And we'll have some more statistics for you on Monday, like the attendance at SI sessions and review sessions by the TAs. And so if you didn't go to any of those and you're going to ask me, hey, how do I improve my grade in this class? There's a lot of resources available to you that you're not taking advantage of. Now, if you got an A or a B, or if you got a C on this exam and that's all you really want is to pass the class, get a C and you're good, then it sounds like you've already found your groove. But if that's not you, and you'll find out tonight or tomorrow, I don't want you to panic. I'd love to talk with you one-on-one. Office hours have been pretty meekly attended, and I'm imagining there will be an uptick after the first exam. There always is. It's routine. Okay, so it's not like doomsday is coming, the grades are horrible. I just feel like there is some room for improvement, and maybe you feel that way too when you walked out of the exam. Okay? Fair? I'd like to believe that I'm approachable. So if you want to talk about it individually, I'm all ears. And if you can't make it to the office hours, we can schedule time. And you all know when those office hours are, right? Tuesdays by Zoom at 11, and Wednesdays in person or by Zoom at 11. And that's because I'm here and I'm available. If you're not available and you need another time slot, we'll find a time slot that works for the two of us. Okay. All right, let's lighten it up a little bit. What did the mom chameleon say to her nervous kid on the first day of school? Just try to blend in, or don't worry, just blend in. I like that. Very well done. Very well done. Pretend, okay, I want you to close your eyes. Pretend you're in the jungle and a tiger is chasing you. What do? Stop pretending. Well done. All right. What? Some of you just got it. All right, I love it. That's awesome. Awesome. I know they're mind benders. There's no question about that. What goes all around the baseball field but never moves? The fence. Well done. Ba-boom. All right. We ready to start our next session? Our next unit? So today and Monday, we're going to get into the skin, or the integument. And this is tracking pretty well with lab right now. All right, I'll wait until everyone's ready. Okay. So we're tracking pretty well with lab, with tissues. Now we're moving into skin, or what we refer to as integument. And this is actually one of my favorite organ systems. I've spent probably 20 plus years in research looking at skin. And that's not the only thing that we do, but it's an organ that's really easy to work with. It's the largest organ in the body. It's easy to access if you're doing research and you want to impact or make a difference. Clinically, there's a huge issue associated with non-healing skin wounds in diabetic foot ulcer patients and venous pressure ulcers and individuals that are older with comorbidities. And if the skin is breached and open, it's a site of major infection. So I get kind of excited about this section. Monday, we'll talk a little bit about some early research, like early in my career, that if you flip through the slides, you'll see some pretty horrific images coming. And I want to tell you that because when I put them up here, they're even bigger. Okay, and you're like, I'm fine. And then I get like 5 people in the back row just, passing out. So I'll give you a warning. You can look at them ahead of time so you can see what we're talking about. There's some pretty aggressive wounds in the later part of this lecture. We won't get to that until Monday. And this is really where we pivot. So the other thing about Exam 1 is, I'll be totally transparent with you. It is a really tough job, Unit 1, for me, to make it interesting. Because it's not probably what you signed up for when you said, I'm going to take anatomy and physiology, and we're going to start talking about glycolysis again, and chemistry, and oh gosh, right? So some of you are just like, oh man, I hope this gets better. This is where it should get better, okay? This is where it starts becoming relevant, but we have to go through the foundational Unit 1, and I promise you, I will point back to it in our lectures, and then hopefully we'll connect some dots. And you'll go, all right, still sucked, but I kind of get it now, okay? So we have to cover that stuff, and now we get to move into the material that maybe is more interesting to you. Things that are a little bit more relevant. They're more translational. They're more clinical. They're more applicable. Whatever that word is that you want to fill in, they're not necessarily basic science that some of you said, I kind of just struggled through it, to be honest. And maybe that's what summarizes Exam 1 for you. Okay, in Exam 2, things are going to be a little bit more like, yeah, this actually makes sense. I can kind of wrap my head around how the skin helps protect the body, how it's a site for vitamin D synthesis, how we sweat, how we thermoregulate, all the functions of skin that we're going to talk about. Maybe it's easier for you to wrap your mind around that versus what's a nucleotide? Okay? You with me? No, that was a bad joke. Sorry, I won't write that one down. I'll tell the kids that one went. Okay. So if we look at skin up here in the upper left, this is a scanning electron micrograph, an SEM. And this scanning electron micrograph, one second, let me share my screen, 1 second. I just broke my rhythm, but I want to make sure that our study tools are pretty dialed in. Okay. So this scanning electron micrograph in the upper left, that's just a fancy piece of equipment that we talked about in Unit 1. So I promised you, here it goes. Do you remember when we talked about light microscopy and electron microscopy? Electron microscopy, we have one in the Weta building. We have a few electron microscopes. If you're interested, I will show them to you. I'm the director of that facility. I'll introduce you to our manager. We'll show you how we make these pictures. It's quite fascinating. Where we shoot electrons at the surface, and they reflect off. We capture them, and based upon their angle of deflection, we can reconstruct an image. And you get these images like a hair shaft diving into the skin, and that's what that's a picture of. Okay, you can take pictures of bugs and all sorts of weird things, but It's a diagnostic tool that's really valuable. And we're going to look at structures like this in today's lecture and on Monday as we look at this organ system that I said is the largest organ in the body. It literally translates from the Latin as covering or to cover. Integument is a covering. So that should make sense. Why do we call it an organ? Why do we call it an organ? Do you remember when we classified in Unit 1 from the atomic level all the way to the organism? There was a test question on this. Why do we call it? It's made up of multiple tissues. It's not just made up of multiple cells, it's made up of multiple tissues to form multiple functions. So a lot of students look at this and say, it's an organ? Like, I think of organs like inside the body, stuff that, swords pierce in some of the movies that I like. And, it's like, it's inside. What do you mean we have the largest organ of our body that's outside and exterior and exposed? Isn't that a bad idea? Well, it has multiple functions, and it's made up of many components or multiple tissues. And I'm going to prove it to you today as we walk through it. Hopefully, I'll prove it to you today. So the two main regions, not tissues, but regions, are the epidermis and the dermis. Epi is on top of the dermis. So the epidermis is the superficial-most layer. Again, back to Unit 1, we were talking about superficial and deep. Superficial is on top, deep is down into the tissue. So the heart is deep to the skin. And the epidermis is superficial to the dermis. And then the hypodermis is deep to the dermis. But the hypo, underneath, hypo being under, hyper being above, so hypodermis is under the dermis, not technically part of the skin. It's not considered part of the skin. Anybody know why? Was it just some arbitrary anatomist that said, we don't include that as part of the skin? Any guesses? It is vascularized, but that's not the main reason. It is made up of connective tissue, but also, again, that's not the main reason. I'd be surprised if you get this. We didn't cover it in Unit 1. You may or may not have covered it in other classes, maybe in high school biology. The answer was, does it have to, kind of a question answer, but the comment is, does it have to do with its function? Maybe kind of how it moves things up towards the dermis. Like, for example, it is well vascularized, right? That's the site that we inject injections into, hypodermic needles. So if you're going to be a nurse, this is the tissue you're going to shoot for. You don't want to inject it into the epidermis, right? It's not vascularized, it won't absorb. You put it in the hypodermis, because of the extensive risk vascular supply, it'll absorb very quickly. But again, its function is not the reason. It has to do with its origin embryologically. So during embryologic development, the hypodermis is actually formed out of the middle layer of the embryo, known as the mesoderm, mesoderm. And the ectoderm, the outer layer of the embryo, is where the epidermis and the dermis are derived from. So they have different embryologic origins. And then they actually lay over top of each other very nicely. But that is the reason that we don't necessarily keep it as part of the organ known as the integument. So it's not technically skin. Now you know why. It is made up of connective tissue, very well done in the back. It is well vascularized. It has unique functions. So you're all correct, but I just wanted you to understand the why behind it. And sometimes understanding the why helps you just say, I don't know, it's just not part of it. Just answer that on the exam and you'll be good. It's nice to know where it's derived from, in my opinion.
Is it a part of a different organ system?
The question is the hypodermis part of a different organ system? No, it's not. It's just considered connective tissue. Yep. Just considered connective. It's a reasonable question to ask. So it's made up of loose connective tissue, both adipose as well as areolar connective tissue, which are the two types that we studied in Unit 1. The dermis, there's a lot of white space on this slide for you to write these facts down, right? The dermis is made up of areolar and dense irregular connective tissue. If it's dense irregular connective tissue, do you think the dermis is strong in multiple directions, or is it strong in one direction? It's strong in multiple directions. Is that important for its function to get through life? Absolutely. What happens if there's a genetic defect in an individual where the dermis is not strong in all directions? Could you see a problem there with wound healing, with bruising? What about if that connective tissue between the hypodermis that's vascularized and the dermis that is on top of it is weak and it fractures often? Subcutaneous hemorrhaging can happen in those individuals. Okay? So, does it matter that you understand connective tissue and the different types? Hopefully you see, yeah, it does matter. Will it be relevant in the future? Hopefully you'll see, yeah, it will be relevant in the future. And sometimes it's just hard to get through it. The epidermis, the very top layer, keratinized squamous epithelium. Does that make sense to you, what I just said? Keratinized squamous epithelium. Define it for me. What does that word mean? What does that string of words mean? Because if you stop the average person on the street and said, well, your keratinized squamous epithelium is looking quite nice today, you might not like the response you get. That is a very complicated string of terms. Yes, let's define it. Go for it. Right there in yellow. Yeah? So that would be the keratinized. Has a lot of keratin and keratinocytes in it. I agree. What about the next term? Squamous? A lot of the flatter cells. Okay, is it stratified or is it simple epidermis? Stratified, does that provide protection? Yeah. Okay, what about the epithelium term? What does epithelium refer to? The type of cell that faces the outside environment, or a lumen. So, we're on slide 2, but I'm going slow because I'm trying to prove to you that this class builds on itself. In Unit 1, why it kind of maybe stunk in your minds, you actually learned something because you're stringing some ideas here together, and I just said keratinized squamous epithelium, and hopefully you understand what we're talking about, and you understand how rich each one of those words is, and it means something very important. They're not just words. They actually have a whole definition, a whole functionality, and that's kind of the elegance of what we're getting into. Okay? 3 words. Slide 2. 10 minutes into the lecture, right? And because I'm really trying to make sure that you guys understand and you're bought into the concept that we're going to build, and the train is going to start increasing in its velocity here quite quickly. What does it do? Some of its functions. Communication. I want you to turn to your neighbor and communicate skin to skin in some capacity. Keep it clean. Keep it clean. Huh? I see some fist bumps. Okay. I see a lot of giggling. Okay. How about just tapping, high fives? Okay. Okay. So, but you read that, you don't come to lecture, you don't watch the lecture, you read that and you're like, I don't know how the skin communicates. I'm completely lost, Dr. Keller. We actually have tactile sensation in our skin. Light touch, heavy pressure, right? Temperature sensitivity. We'll get to all that. But you're communicating, you can communicate with your outside environment or with another individual through the skin. Site of waste secretion through sweating. Protection is probably the most obvious. Protection as a physical barrier against infection, water loss, abrasion, UV damage. So all of those are under protection. I'll say them again. Physical barrier against abrasion, water loss, microorganisms or infection, UV damage. Now, many of you kind of grew up in a society where it was, highly antimicrobial soaps, or, during the pandemic, we were hand washing like crazy. How many of you just, you compromised the antimicrobial for just cracked skin, right? Because now you're drying out your skin with all the hand washing. And so a lot of overuse on antimicrobials can actually be deleterious to us, right? There's a natural bioflora that lives on our skin. And it's one thing not to wash your hands after going to the bathroom, but it's another thing to be chronically washing your hands because you're removing a lot of those microorganisms that live in symbiotic relationship with you. So in fact, there was a CDC study that was done in Chicago that actually showed no difference between groups who used regular soap and those who used an antimicrobial labeled soap for 1 year. Okay, this was pre-pandemic. This is back in the 90s. But I just wanted to share with you kind of this philosophy and give you a moment of caution with respect to how much we overdo hand sani and hand washing. We have to be a little careful. Question?
So, when you get tattoos, they usually tell you to use some sort of antibacterial soap. Would that, the same kind of thing as is there a difference in that compared to using regular?
Question is, when you get tattoos, they encourage you to use an antibacterial soap. Why is that? Well, with a tattoo, you're actually breaching the barrier function of the skin. And depending upon how big the tattoo is, now you're at huge risk of infection. So that's not a daily thing, I would imagine, okay? Even if a lot of you have a lot of ink work. And we'll get into tattoos. We might get into it today, depending upon how quickly we can kind of hit our pace, okay? Other functions of skin. Temperature regulation, hot versus cold. If you're hot, that vascular supply will actually vasodilate, bring warm blood from your core, middle of your body, to the surface, and that warm blood will allow for a sweat response and evaporative heat loss. If you're cold, you're going to vasoconstrict, and you're going to actually shunt blood away from the skin so you don't lose a lot of heat. So temperature regulation in the skin is based upon blood flow. Vasodilation or vasoconstriction. Vasodilation if you're hot, vasoconstriction if you're cold. Vasodilation when you're hot, that's why you look flush. A lot of blood is flowing to the skin surface, and so you have more of a red color. Okay? Vasoconstriction in cold environments, and then we'll talk a little bit about goosebumps or what happens with the erector pili muscles when you get cold, okay, and how they're basically flexing in a way to try to preserve heat loss. Yes? So the question is, why does your face turn red when it's cold? Well, it just depends on the environment. If there's a lot of wind, you might have some wind burn going on. If you're cold but yet exercising, like you're cold but yet walking, there may be multiple areas in the body that have different types of vaso activity. So vasoconstriction and vasodilation doesn't necessarily mean it happens universally across the entire body. Okay, so you could be exercising, your muscles are warm, other parts of your body are cold. Okay.
So when people work out and some people get more red than others, is that just because they're faster, that's because their blood flow faster?
Yeah, when people work out, like the different levels of rubor or red color, a couple things that go into play. One is circulation, okay? The second one is thermoregulation, how well your body is thermoregulating, and everybody thermoregulates at different levels. And then third, the visibility of a redness is going to depend upon the tone of the skin too. In more fair individuals, you're going to be able to see more red than you are in individuals that have darker colored skin. Okay. Metabolic functions. So when we say that we've got multiple tissues that do multiple functions, we've listed quite a few functions already. Let's repeat them. Shout them out to me. What are some functions of the skin? Protection, communication, waste secretion, okay, thermal regulation, and now metabolic functions. This is a really interesting image. This is an en face view. That means That's Latin for face, a face view. So it's kind of like a selfie. Okay, bad joke. Sorry. En face view of skin, looking down at the skin, and what you see are like tiles. And I want you to be able to appreciate the squamous nature of the skin, where this is like a flat pancake. Some of them are nucleated, some of them have an appearance that they're not nucleated. Or I should say, some have an appearance of nuclei, some of them appear that they're not nucleated. Because they're so flat, you're seeing multiple layers, and they're translucent, you're seeing actually through them. So this cell that looks like it has a nucleus is probably deep a few layers, like in the spinosum layer. Make sense? And they're flat, and they have this connection in between them, but this is a wide surface area. The cells are spread out widely. And the metabolic activity here is the production of vitamin D. Vitamin D is critically important for calcium absorption. And so you've heard a lot of people say, well, the sunlight is really good. Go out into the sun and synthesize vitamin D. So what is happening? Well, you have multiple organs that are involved. The multiple organs that are involved, it starts with the skin. And then it moves to the liver, involves the kidney, and then has activity on the bones as well as the gut. And so if you follow this chain of events, we're looking up top here. Let me back this up so we don't get too confused. We're starting up top in this flow chart, 7-dehydrocholesterol, which is found in the skin. Exposed to sun, it turns into D3, or vitamin D3. Cholecalciferol. So if you look at your multivitamin, you look on the back and it has calcium supplementation, you might be taking D3. Okay, that comes from the conversion of this cholesterol molecule to D3. That's the intermediate. And then you see this enzyme, 25-hydroxylase in the liver, that converts it to 25-hydroxy vitamin D. And then you have another enzyme in the kidney, 1-alpha-hydroxylase in the kidney, which turns it to 1,2,5-dihydroxyl vitamin D, or calcitriol. And then from calcitriol, we actually utilize it to bind to numerous different vitamin D receptors. So let's look at this figure that I threw up here. We've got cholesterol molecule, which we studied in Unit 1. We looked at some of the architecture of some of these molecules. And we go from cholesterol, 7-dehydrocholesterol, in the presence of UV light to vitamin D3. The liver converts it to calcidiol. The kidney converts it to calcitriol. Calcitriol has activity on bone, and it has activity on kidney as well as gut. So it influences boredom resorption, right? It reduces the excretion of calcium in the urine, and it increases the absorption of calcium and phosphate in the gut. So what you're doing is you're actually making calcium available. And that can be very, very valuable for bone development. So this is the link of why sunlight is valuable. Now, you can get it out of your food. You can see on the right side there's a way for you to insert into this mechanistic tree using an intermediate out of the food that you eat, which is vitamin D2 instead of vitamin D3. Make sense? Another function of the skin for metabolic activity and calcium management. What does the hypodermis do? It has a functionality. It's not just a fat layer. Well, it helps with movement. It allows the skin to move freely. You can take your skin and it shifts a little bit. It's nice if you come around a corner a little tightly and you rub up against the wall. You know, you don't like tear your entire skin layer off. So there's movement capability, shock, insulation. It's not necessarily present everywhere. You can see on this schematic diagram the areas of higher concentration of hypodermis. So for example, none of us have really any hypodermis on the tibial crest or our shins. Right? It's pretty much thin skin stretched over top of bone. And if you fall in a space that doesn't have a lot of hypodermis, you can see bone real quickly if the skin splits. So it's not necessarily present everywhere. Okay, the skin consists of the epidermis and the dermis. We talked about this, so I think we'll kind of skip over it because I already did it. But why do you think that injections are delivered into the hypodermis rather than the epidermis or the dermis? All right. I didn't think we were going to get discussion on it because we kind of covered it, but that's cool. You guys want to talk? That's good. So tell me, what did you discuss? State of the Union address last night, or did you talk about this? Yes. It's more vascularized, heavily vascularized. The hypodermis is heavily vascularized. So if you see in this diagram, this is like a little cubed cutaway of skin. And you've got the epidermis, and where this is kind of peeling off, you see how there's these kind of like ridges? That's the epidermis that this figure is looking to pull up, and it's leaving the dermis down. It's not that the dermis is avascular. The epidermis is avascular. The dermis has small vessels, small capillaries and venules, but the much larger vessels are here in this fat layer. You see this yellow fat layer at the bottom? Globular-looking oval structures. That's where these larger vessels actually live. So from an injection standpoint, delivering a hypodermic injection is going to get faster absorption into the bloodstream. Okay, we're going to focus on the epidermis for a little bit, and then we're going to focus on the dermis. And I got couple quiz questions for you, and then I think actually we might dive a little bit into material beyond today, kind of get ahead a little bit for Monday. So with the epidermis, we've got two main types of skin that I alluded to just a moment ago. We've got thin skin, which covers most of the body, 4 layers, and we'll go through them, about 75 to 150 microns thick. And the layer that it's missing, what's the layer that it's missing? Those of you that have been at lab. Stratum lucidum. So did you hear about where this came from? So strata means obviously layer. Lucidum, where's this origin in the Latin? Lucidum. Light. Light. Like, Lucifer was considered the angel of light originally. That's where the name derives from. So lucidum layer is this light layer, and I'll show you pictures of it. In contrast, we've got thick skin, has the stratum lucidum layer, and on histology, you can really tell the difference with the lucidum layer. You should be able to see it. It's pretty obvious. And I'm going to show you a couple of cool things today where we're going to look at histology in 2 different ways. We're going to look at it in an old school way, and we're going to look at it in a new school way. And I'll explain. Palms and soles of the feet, 5 layers, much thicker, 400 to 600 microns thick. Has the stratum lucidum layer that we'll look at that's very, very obvious. If we look at them against each other, the stratum corneum of thick skin is much more pronounced than the stratum corneum of thin skin. And so thick skin of palms of the hand, soles of the feet, obviously lots of pressure, right? And some of you have calluses, right? You do an activity over and over and over again. One of my daughters, I think I mentioned, is a aspiring ballerina. And her feet, I love her to death, but her feet are disgusting. Okay, I'm just going to be honest. Okay. It's the only part about her that's disgusting. I love all of her. Her feet, I could do without. And the thickness of some of the calluses, and I mean, she's 18, 19, has bunions. I'm like, what? Like, that's like an old person thing. But she's like, no, Dad, look. And I'm like, I don't want to look. I believe you, hon. I don't think you would lie about that. Okay. So, thick skin found in areas where there's a lot of pressure, a lot of repetitive injury, okay, trauma. And that's why you build up a callus. Some of you have them on your hands. Some have them on your feet, like my daughter. Some of you have them on maybe other places. If you play a sport and there's a piece of equipment that rubs on that area, you'll develop calluses in those areas. So now I just took the coloration out of the image. Okay, so it's color. Now it's black and white. And the tool that I want you to start thinking about here is, can I start identifying some of these structures if I don't see the coloration of the stain? And the reason this is interesting is, if you look at historic medical journals and scientific publications with histology, like from the 1800s and the early 1900s, all of the histology is done in black and white. And all the conclusions were drawn from black and white histology, because you can actually appreciate architectural shapes and different features and patterns. And so that's what we're going to do. We're going to start looking at the different layers of the epidermis, and I'll have a colored image that you're used to seeing in all your textbooks, and then we'll take a peek at the black and white image and see if you can identify some of those shapes. Because on the exams in the future, if I change the coloration around, if the stain looks different, it's amazing how it throws students off. And if you're focused on shapes and architecture, we actually might be doing you a better service by preparing you in that way. Okay, so let's talk about the stratum basale. So we're going to go from deep to superficial. Stratum basale, basal layer. The basal layer. This layer is closest to the blood supply. It is considered the regenerative layer of the skin. This is where your skin turns over. Your skin regenerates when you need to heal, but your skin also regenerates just in a normal process. About every 6 to 8 weeks, you have a complete turnover of your skin. So you think about reptiles, snakes. You've ever seen a snake shed its skin? It's pretty gross, right? We do the exact same thing. We don't molt. We just do it a little bit every day. We just drop a little bit of us everywhere we go, leave it here, dust it over there, right? It's kind of gross, actually. At least they have the courtesy of like leaving it in one spot. We just shed everywhere, like on the seats that you're sitting in. You're like, ooh, gross. Like, yep. So the stratum, that's the stratum corneum layer. That's the superficial most layer. But the basale layer is where the cells are the most round. We've got keratinocytes. We have melanocytes. We have Merkel discs. Melanocytes produce melanin. We've already talked a little bit about that. We're going to go into that in more detail. They allow for skin pigmentation with melanin, and it helps to protect us from UV radiation. The role of melanin as a pigment isn't to make us look tan. It's actually to protect us from UV radiation. That's the value, the physiologic benefit of melanin. Those come from melanocytes. Merkel discs. These are sensitive to pressure. These are tactile discs. And that's denoted by these arrows right here that are found at the level of the stratum basale. I've colored it here in yellow on this slide so you can kind of appreciate where we are, but we're at this basalmost layer of the skin. So if I remove the coloration, here's the color removed, it's this first row of cells that are slightly arranged in sort of in parallel fashion to each other. Can you appreciate that? The regenerative layer. Stratum spinosum. This is the next layer up. It's been pseudo-colored in orange right here so that you can kind of appreciate where it is in relationship to the layer beneath it, which is the stratum basale. If I take the coloration out, it's this arrow that's pointing to this big, thick region of the stratum spinosum. There's still some cell division happening. The cells are starting to squish. Now remember, you know the story. It's going to go from a round cell to a flat squamous cell as it works its way up. And along the way, it's going to start spitting out organelles. Spits out the nucleus, like out it goes. Okay? Once the nucleus and organelles are gone, it can start flattening even more and more and more. It allows you to stack it. And remember, you want a stratified epidermis up top for protection. So you want it stratified, and we don't want to have skin that's like 3 inches thick. So we take the cells out, and we squish it down like armor. It's like scales on top of us. Stratum spinosum, still some division. This is where you find dendritic cells. Now, this confuses students. Dendritic cells, that sounds like a dendrite. That sounds like Unit 1 when we talked about neurons. I know it does, but they're completely different. Dendritic cells are cells that are part of the immune system. Now, think about this. You're like, what is this immune system cell doing in the skin? In one of the deep layers of the skin, the stratum spinosum layer. Any ideas what immune cells known as dendritic cells, what do? They find foreign objects or substances, they phagocytose them or engulf them, and they display them to your immune system and say, go attack this. Why would you want that here in the stratum spinosum layer? Sort of this lighter layer between sort of this pinch point and this darker band right here. Why would you want those there? Oh, sorry. How about we let someone up front go? Because you already had an opportunity. Is that cool? What a gentleman. All right, I love it. All right. The answer was an assumption that if you cut yourself and bacteria gets in, you're going to want a line of defense there with a specialized cell to actually engulf it and display it to the immune system. You'd be absolutely correct. I mean, this material isn't that complicated, right? It makes sense. Okay? So that's exactly what these dendritic cells do. They're called antigen-presenting cells, APCs. Now, you find them in the lining of the nose, the lungs, the stomach, and the intestine, not just the skin. That should make sense to you. All the areas where you're exposed to the outside environment, you're going to find these dendritic cells. Okay, stratum granulosum. What does this word sound like, granulosum? Layer that is what? What's that? Little grain. Did you say grain? I couldn't hear you, I'm sorry. Yeah, you were correct. So it's granular in appearance. So this is the layer, the stratum granulosum is 3 to 5 cell layers thick. So it's a thinner layer. You can see it here in green. I'll take the color out here. It's this band right here that is just beneath sort of this little invagination as you're making your way up towards, the superficial aspect of the skin. So here it is in, Here it is in green, right here. And then here, it's this layer that just follows that shape as the folding of the skin is taking place. This is the layer where you've got lipid-filled vacuoles that are exocytosed. We talked about exocytosis and endocytosis in Unit 1. What does exocytosis mean? When these cells are exocytosing vacuoles, what are they doing? They're releasing them. They're getting rid of it. Remember, I told you to go from a round cell to a flat cell, you got to get rid of stuff. You're dumping contents. It happens in the granulosum. So little grains, granular in appearance, because they're actually degranulizing. They're actually removing the contents within them. And as it does this, you get a lot of lipid outside of the cell that creates a waterproofing feature at the same time, which is kind of cool. So now you're waterproofing this thing with like a Gore-Tex covering while you're getting rid of the contents so you can smash it down into a squamous flat shape. I think it's pretty cool. This is the barrier between the outside environment and the inside environment. The dendritic cells are deep to this. They're inside of you. This granulosum layer is kind of that barrier between outside and inside. And things will not penetrate past this layer because of the lipid aspect, unless they're fat soluble. right? Certain topical fat-soluble substances will absorb, like estrogen, for example. So how can you deliver an estrogen patch on the surface and it absorb through the skin? Well, it's lipid soluble, so we can get through. So some of your patients, if they're on hormone replacement, they might have an estrogen patch. And you're like, how come I'm not injecting it? Don't I have to go into the hypodermis? Well, no. If you have something that's fat soluble, it can get through the membrane. And we covered that last unit in Unit 1. Okay, stratum lucidum. This is our devil layer, right? I love to talk about this. But people don't forget it. Layer of light. So it's a light-colored layer. It's only present in what type of skin? Thick skin, well done. It's highlighted in blue right here, but if I took this color out, it's this layer right here that's shadowed right above where we were, and it's almost like it's highlighted. It's avascular, it's anuclear, right? So there's no organelles or nuclei in this layer. It's the first layer absent of cell organelles, and that's why it has a light appearance. Like on light microscopy, light passes right through it because there's nothing inside the cell. So the cells are now dead. The stratum lucidum layer, some of you are like, oh, I like that one better. The Lucifer layer is dead. Okay. The stratum lucidum layer looks light because in color you can shine light through it and nothing is refracted off of it. There's no cell contents, no organelles, no ribosomes, no mitochondria, no nuclei. Everything's been spit out in the waterproofing that we just went through in the granulosum layer. Last one. What is it? Stratum corneum. Superficial-most layer, packed keratin. Keratin is a protein that gives structural strength or integrity to tissues. So the skin, I said, it renews every 6 to 8 weeks. You actually slough off a pound of stratum corneum every year. A pound of stratum corneum, on average, an individual sloughs every year. It's completely keratinized, this layer. And this is where the cells go through a process known as exfoliation. Right? Some of you think, oh, well, isn't that like what my mom does at the spa? Like, well, the whole concept of exfoliation, I mean, the day spas borrowed it from science. It just means to eliminate the layer of cells. The removal of the oldest cell and the dead cells superficial, allowing for newer cells underneath it to replenish it. You do it naturally, but some of these techniques of scrubbing and face scrubs and different types of brushes and sanders or whatever you use, right? Help to exfoliate or remove those dead cells more quickly. But you'll do it naturally, 6 to 8 weeks. They shed consistently. It's not like all at once. It's not like we talked about with molting with reptiles. This happens on a regular basis. And it's this top layer that's kind of colored in purple. If I take the color out, it's this 30 or so layer, thick layer of stratified squamous epithelium as we move superficial. So if we look at them all together, this is the pseudo-color here. Here's just a standard H&E. I want you to be able to appreciate as we move from superficial to deep, or when we go from deep to superficial. Okay. I want you to talk about this with your partner. Just someone who's sitting next to you, just group up. And I want you to discuss, why do we have differences in skin color? Why are they there? Why is there a variety in this room? All of us have different tones, skin tones that exist. So talk it amongst yourselves based upon what we've covered so far, and let's hear some ideas. Okay, thoughts. Let's get someone to enlighten us. What do you think is the difference here? I see someone. I'm going to go there. I'm going. All right. Difference in skin color, skin tones.
The difference in skin color is the amount of melanin that is synthesized. So if you're white, you still have this same amount of melanin, just it's not synthesized.
Everybody hear that? So the amount of melanin that's made. So that's absolutely correct. So melanin is a pigmented protein, brown in appearance. I think in one of the early lectures I showed you some histology of tissue that was unstained. It was clear after we processed it. And the melanin you could actually see as kind of little dots of brown. And we all have about 1,000 to 2,000 melanocytes per square millimeter of skin. But the activity level of those melanocytes differs in all of us. Nobody has the same exact skin color. Nobody does. And it's not necessarily the quantity of melanocytes, but it's actually their productivity and how much melanin they make that gives us kind of the variation that we see in the world. And the purpose is for UV protection. And we're going to talk about that. We might get to it today. It may be Monday that we talk about that. So if you look at a color map of skin across the world and you consider locations equatorial in nature, closest to the equator, that have the most intense sun and the most amount of sunlight basis around the year. And then if you move more north on the globe or more south on the globe, you start getting into regions where there's less sunlight. So any folks from Alaska in the room? Okay, how much sunlight's happening right now in Alaska? Very, very minimal, right? Very minimal. So if we look at indigenous people, so this is indigenous people, where, some of the original people in those regions, the skin color tends to correspond with the amount of protection that's necessary based upon the environment, which is quite interesting. Now, of course, we've migrated all over the place, but vitamin D and skin color, there's a correlation, right, which is kind of interesting, where if you were living further from the equator many, many years ago as an original people in that area, the amount of sun that you're going to get is limited. So if you had more melanin production, it would not be advantageous for you based on the amount of vitamin D that you can manufacture. Make sense? The power that you need from the sun and the energy in UV radiation is important for vitamin D synthesis. Now, if you live equatorially, you have exposure to a lot of sunlight, you don't need much sun, or you're getting too much of it. So your melanocytes are going to produce more amounts of melanin in order to allow for that protection or that protective nature. Make sense? All right, dermis. That was the epidermis. That's the superficial-most portion. Now we get into the dermal layers. And we're going to talk a little bit about the characteristics of the dermis in the remaining time. Well, it's very strong. It's a very flexible material. It's made up of mostly dense, irregular connective tissue. But we have a number of different cells and molecules like structural proteins, collagen, elastin, reticular fibers, fibroblasts, macrophages. Fibroblasts are cells that live in the dermis that manufacture collagen and a lot of extracellular matrix proteins. And macrophages are found in the dermis. They're similar to the dendritic cell that you found in the epidermis. But macrophages are your workhorse cell for inflammation. So in a wounded site of skin, your macrophages will be the ones that gobble up the debris. They phagocytose the bacteria or the viruses that actually make it through the compromised skin. This is all found in the dermal layer. It is vascularized in comparison to the epidermis. It's highly vascularized. In comparison to the hypodermis, it's moderately vascularized. And we have many sensory receptors in the dermis. So what I'm looking at is this layer, and hopefully you can appreciate this looks like dense, irregular connective tissue. Do you see all the shapes moving in many different directions? They're not aligned fibers. They're not straight. They're all over the place. And this layer that's highly cellularized is the epidermis, and the layer up here that has kind of these little wisps, what layer is superficial that is sloughing off cells that are dead? What's this layer up here? Stratum corneum. Okay? So I should be able to ask specific questions on histology about different layers and what's present, correct? Here's another picture. We've got the epithelium up top denoted by this bar, and then we've got our dermis that's denoted by two different layers. We call this a papillary layer that's closest to the epithelium, and then we call this a reticular layer, which is more deep. So the purpose of the epidermis: blood supply to provide nutrition to the epidermis based upon diffusion. What was the diffusion distance of oxygen? Do you remember? In Unit 1? Anybody? It's about 250 microns. So if you move 250 microns superficial from the dermis, you're at the threshold of where oxygenation is compromised. That's right about which layer? Stratum? Any ideas? The layer that's the dying layer. Granulosum, where they're starting to lose all their contents. That's right about 250 away from the dermal layer, where the vascular supply is. And so that's the stimulus to say, okay, let's start losing organelles and shutting things off. We're going to squish the cell and create a dead cell because of that distance away from your blood supply in the dermal layer. Sensory perception, structural integrity of the skin. The layers that make up the dermis. We have our papillary layer and our reticular layer. Papillary layer, closer to the epidermis, areolar connective tissue, and what we refer to as the dermal papilla. The dermal papilla are these little extensions or these projections that kind of look like a, you know how, it's not called tongue and groove, I'm not a carpenter, but if you have a really well-made dresser and you open up the drawer and you look at the edges, they're actually like, maybe it's called a dovetail or something. Does anybody help me out here? Who does carpentry? Dovetail, thank you. So you know that it's a better made drawer. You can go home tonight and be like, oh yeah, I got it at IKEA, it's super cheap. Okay. But the dovetail, where it's kind of like interlocking, that adds strength. I don't know, it was like an answer from the audience in the back. I don't know who answered dovetail back there, but it's stronger. That's why we do that, right? So this is a stronger layer because the dermal papilla go up, and these projections of epidermis, known as the ready pegs, come down and form an interlock. And that's part of this papillary layer. And then down low, deeper, is the dense irregular connective tissue that we've been pointing out in the reticular layer. So one more picture to kind of highlight this. Here's our dermal papilla right here. I really want you to be able to appreciate kind of this interlocking strength reinforcement at the level of the skin between the epidermis, superficial, and the dermis that's deep. And this is the dermal papilla, this part that's projecting up. And that's part of this areolar connective tissue in the papillary layer. Down here is our dense irregular connective tissue as part of our reticular layer. If I take the coloration out and we just look at a black and white image, does this look more dense to you? Yeah. Do you see how much lighter this is or open this is compared to down here? And this is the epidermis as it projects downwards, trying to meet the derma papilla as they project upwards for strength. Clear? Sir?
So for the papillary layer, if it's made of areolar tissue, how come it's hard to really see the fibers?
How is it what?
How come it's really hard to see the fibers in the areolar tissue?
Because the magnification. Yeah, so the question was, if this is areolar connective tissue, why can't I see the fibers? We're just at a higher magnification view. We're zoomed out. If you zoom in, like what we were looking at in Unit 1, you would see those individual fibers. That's a really good question. So in lab, you're going to be switching from different objectives. You'll be able to appreciate different features depending upon your magnification and how close and how far away you get. Make sense? What else is found? Kind of as we wrap up this portion of skin, what else is found? Well, hair follicles. We introduced the lecture on hair follicles, where the epidermis actually dips down or invaginates in and actually houses the hair shaft and allows for the hair follicle to grow and extend out of the epidermis. And you can see this happening here in a histology image, and you can see it highlighted on the far right in a cartoon or a schematic. We also have sweat glands that are found here for thermoregulation, allowing for that blood supply to be brought to the skin, to the gland itself, and sweat is filtered blood. That's all it is. It's filtered blood. So when you vasodilate, you mean more blood flow goes to the skin. You filter that blood, and it's basically a filtered plasma that's made up of sweat. So you're eliminating waste products that came from the bloodstream before the kidneys had to do it. You'll learn about kidney dialysis next semester in 202. But here, you're seeing how you're eliminating and secreting waste products through sweating, not just through other organ systems. We've got a vascular supply that we've talked about. You can appreciate the vascular supply both in the hypodermis as well as in the dermis. And we also have nerves that live here in the dermis. You cut yourself, it hurts. We have these erector pili muscles. And the erector pili muscle is actually, or piloerector muscle right here, erector pili or piloerector, depending upon the textbook that you're using. This allows for control of the hair follicle or the hair shaft itself. And so when we get cold and we have goosebumps, right, or chicken skin or whatever you call it, it gets its name in effect from geese, and that are goose feathers that are grown from stores in the epidermis that actually resemble human follicles. But piloerection is the standing up of that hair in your skin, and it's thought to be more of a vestibular characteristic or a remnant characteristic, because in avian species, it's a way to fluff the feathers to create dead airspace to capture warm air. It's like wearing layers in winter. And so obviously we don't have feathers and we're not hairy enough for it to really matter, but that is a feature of piloerection that still exists. And so these goosebumps or piloerection is sort of one of these characteristics that biologists kind of point to say, hey, this must be a remnant characteristic that we don't necessarily use anymore, but it's something that's similar in a lot of our cousins, biologically speaking. Okay, let's look at a couple review questions. So, consider where keratinocytes first appear. In the epidermis, and then answer this question. Which stratum or layer, that's what that word means, is the first to contain dead or dying cells? Use the letters. Which layer do you think it is? Let's hear it. Let's go backwards. E, D, C, couple, B, A. So what is the layer called by name? Stratum granulosum? Stratum granulosum? Very good. What would you label that? B? I would label that B. Okay. How about this one? The papillary layer is labeled what in this picture and is made up of what tissue? Papillary layer is labeled what in this picture and is made up of what tissue? Now on the exam, I'm not going to have G options, but, okay, so I'll have to shorten this down a little bit. All right, first volunteer. The papillary layer is labeled what? Two and areolar. Okay, great. Perfect. E. Well done. All right, what I want to do is I want to start, we have a little bit of time, I want to start this next section that we're going to cover on Monday. We're going to move into skin derivatives. And start beginning to talk a little bit about other things that are basically found in skin, like hair follicles, sebaceous glands, sweat glands, and nails. And we're going to start diving into these accessory structures or these derivatives that are found within skin. So, hair, what do I need to know about hair? Well, we have three different types. We've got lanugo, vellus, and terminal. So let's go backwards. Terminal first. Terminal is coarse, long, pigmented hair that's found in the scalp. Right? So it's the hair on our head. And vellus is fine, unpigmented hair found mostly on children, women, and in areas in men that the hair doesn't really get dark. And lanugo is hair that typically is found on the fetus and then is actually shed as the infant gets a little bit older. We have three zones of keratinized cells, right? So hair is mostly keratin, and that's why it's so strong. We've got the bulb region at the base. We've got the root, which is kind of a connective tissue sheath that surrounds the hair shaft. And then we, of course, have the shaft itself. The three layers to hair, if we look at microarchitecture or the microanatomy, we've got the medulla, the cortex, and the cuticle. So the medulla, loosely arranged cells and air space in the middle. Cortex surrounds that. Densely packed keratinized cells. And then we've got the cuticle. And the cuticle is a scaly outer covering, the outermost part of the shaft. And it's a hard shingle-like structure. Like think of roofs of a shingle where they're kind of laid on top of each other. Gives it strength, protection, as well as waterproofing. Now, there's some correlations to different structures biologically, like horns in animals that are mostly made of keratin. And let's talk a moment about gray hair. So why does our hair change color as we get older? When I was a younger guy, this seemed like it was so far away, but now, it's happened. So what happens is the pigment stops being manufactured. And the clinical term, vitiligo, is gray hair. And it's where the hair loses its pigmentation. You have a decrease in the melanin and pigment production in hair. And so you actually have a shaft that's absent of color. And what you're seeing is mostly air. And that's why it has a light white or gray appearance to it, because the coloration is no longer being manufactured. Kind of interesting. So if we keep going and we look at nails, these are basically modified epidermis. Modified epidermis. We've got three parts. We've got the root, the body, and the free edge. The root is the progenitor or the regenerative layer. If you've ever lost a nail, it grows back from the root. And as long as the root bed is not traumatized or damaged, you should get the nail coming back. Now, if it's traumatized or damaged, you may not get the nail, or you might get kind of a weird variation as it grows back. We've got the body itself, which is kind of what you paint. And then we have the free edge, which is what you trim or shape. And because there's underlying vasculature, clinically, we can actually get really great oxygen saturation measurements through the nail. And those are those little units that you put on. And we can also sense, even before that, if the patient is cyanotic or cyanosis, meaning they're cyan is a blue color, right? So if the nail beds look blue or cyan in color, you don't even need an oxygen sensor. You can tell that the patient is actually low on oxygenation. If we look at a couple of different glands, sweat and sebaceous, these are discriminated by or differentiated by in different categories. Sweat glands, we call them sudoriferous glands. We've got three types: merocrine, apocrine, and modified. And then we've got a term known as insensible sweat. Insensible sweat is basically you're always losing a little bit of perspiration all the time. And that's part of your evaporative loss of water and why we will dehydrate if we don't consume water enough in our diet. Merocrine glands are the most common. All right, they're found on the palms, soles, and the forehead, especially. They're high populations in those areas. And they secrete via exocytosis. They release onto the apical side. I told you sweat is a filtrate of blood. So is it possible clinically to have blood in your sweat? Yeah. And it actually has happened in very extreme circumstances of stress, where patients will actually rupture their sweat glands and have actually blood coming out of their pores. It's quite crazy. Okay, we're going to pause here. We're going to pick it up with this slide on Monday. Okay, have a nice weekend. You all have a quiz due on Sunday night. I'll send out an announcement. A quiz on chapter 5.